So the last thing to talk about in regards to protein structure is the regulation of protein structure and number. So when we talk about proteostasis, we're just talking about the fluctuation in the number of copies of a protein. So later on we'll talk about how proteins are made, but you probably know that they're synthesized by ribosomes that are reading an RNA sequence. And so that's where all proteins come from. And then once they are formed, and that means once the amino acids are connected together, then they go through the process of taking on whatever their final or functional shape is, what we call the native form or native structure. And so as it's coming out, we call it the nascent polypeptide. And then as soon as it merges, it becomes, or it's called an intermediate form. So proteins, if you just throw any amino acid sequence into a solution, it will fold into some sort of shape. And sometimes that folding intermediate or that initial sort of form is the final form. But the folding process eventually, one way or another, turns into the what's called the native protein, which is the protein form that actually carries out whatever function it's supposed to do. It's the functional form of the protein. But sometimes the folding process can have mistakes, and so you end up with a misfolded protein. So a misfolded protein is just another three-dimensional conformation of the protein that is somehow non-functional, meaning that it doesn't have the correct shape. And so this can happen during the folding process, so the folding intermediate can turn into a misfolded protein. Sometimes the native protein can become unfolded, and then you end up with a misfolded protein. Both native and misfolded proteins eventually get broken down. So there's a whole process of protein degradation carried out by this protein complex called ubiquitin proteasomes, and these eventually take all proteins and break them down. So most proteins have sort of a shelf life before they are degraded. And of course, then new proteins are made. That's why we call it proteostasis, because the number of proteins in a cell most of the time is kept relatively constant. So as new proteins are made, old proteins are broken down. And this is especially true for, again, misfolded proteins. So if you have a misfolded protein, a couple things can happen to it. Either it can get refolded, so there are complexes called chaperones, which we'll talk about in a second, which can take misfolded proteins and convert them back into an intermediate form, and then allow the intermediate form to become native proteins. Chaperones are also involved in turning, or can be involved in folding from intermediate to native proteins. And then, but then the other thing that can happen is these misfolded proteins can turn into other protein complexes, or the folding intermediates can turn into these complexes where you can have these aggregates, so proteins can just clump together into a big tangled mess, or they can turn into these things called amyloids, which we'll talk about in a second. But usually those also get broken down. So these complexes of misfolded proteins eventually get broken down again into shorter fragments, and then they get reused or recycled. The amino acids get recycled. So this is sort of the ultimate fate of all proteins. Either they get folded correctly and then they carry out their function and they get degraded, or they get misfolded, and then if they get misfolded, they either get refolded into their correct form, or they get broken down and recycled. So again, this is just sort of a general property of how all proteins work. Again, to make very clear, protein structure is what determines its function, and we know this in part because if you denature or unfold a protein, denaturation is basically just a word for causing a protein to leave its native shape and unfold into another form, then that protein will lose its function. There are lots of ways that lots of things that can lead to denaturation of a protein. One is temperature. So if you heat up proteins beyond a certain point, then they will unfold. Basically, they sort of melt. And in fact, we call the point where the temperature at which a given protein becomes about 50% melted, its melting temperature, or the TM. And different proteins have different TMs, different melting points, but all of them, we know, have the property that when they become denatured, or as they become denatured, they lose their function. So this is true for enzymes, it's true for structural proteins. It doesn't matter what the type of protein is. If it becomes denatured, it won't be able to carry out its normal function. And so again, there's other ways to do this. Another way is if a protein has a lot of disulfide bridges, those disulfide bridges again can be broken by reducing agents. So things like urea and mercaptoethanol will cause the reduction of those disulfide bridges. So this is an experiment where they took a protein which had disulfide bridges, reduced those, and again that causes unfolding of the protein. And when you do that, again, what you do is you lose the function of the protein. So if you try to assay protein activity after denaturation, you can see that it goes away. But if you remove those reducing agents, so if you remove urea and mercaptoethanol, then eventually, it depends on the protein, but it can go back to its original native state, and the catalytic activity or the enzymatic activity, whatever it is, can come back. And so that's evidence that, again, the shape of the protein is really what determines its function. And preventing denaturation or making sure protein gets into its correct shape is crucial for its operation. So the folding of proteins, again, happens as they're made, but the process is kind of stepwise. So in other words, the protein doesn't just fold up all at once into its final form. You usually see regions of the protein kind of folding in a stepwise fashion. So this is an example where, for this 56 amino acid peptide, you have first the sort of residues here, 8 to 15, and then 6 to 17, eventually forming these beta sheets. And then in the middle, you have the region between 20 and 39 forming an alpha helix. And then towards the end, the residues 43 to 54 forming another beta sheet. And then the beta sheet and the alpha helix form a motif. And then the two sets of beta sheets form a larger beta sheet. And then that's just how the protein folds. But if you disrupt any one of those steps, you disrupt the folding of the entire protein. And again, that also depends on the sequence. So if you change the amino acid sequence, you will change the shape. Again, what determines the native shape, what determines the ultimate final form of the protein, has to do with its energy state. So these proteins are folding as molecular bonds are rotating, and functional groups and other parts of the protein are either maybe sterically interfering with each other, or creating hydrogen bonds, or creating new hydrophobic effects. So that means that there's lots of different shapes a protein can take, and each of those shapes has a particular energy state associated with it. And but usually for most proteins, there's one again ultimate low energy state, which is usually the native state. So the process of a protein folding into its native state is kind of its process of going through these different stages, trying to find that native state. So some proteins, and these little funnels just kind of are a way of depicting the energy state of a protein as it goes through its different conformations. So some proteins, like in this situation here, you have the protein going through lots of different gradual pathways. So in other words, it sort of gradually folds, and it can start from lots of different starting points, but ultimately it's going to end up in that same native state. This is much more common, though, or can be more common, where you have multiple energy states, and some of them actually can be somewhat intermediate, or some of these intermediate states can be somewhat stable. So you can see some little peaks and valleys here, little shelves or plateaus, where sometimes the protein can fold into these intermediate shapes that are not quite as stable, not quite as favorable as the native state, but maybe they're somewhere in between. And so those would usually be a misfolded protein, and sometimes that's how proteins end up in their misfolded state. Other proteins are much more simple, so there may only be kind of one native state that's even remotely energetically stable, and everything else is completely unstable. And so there really are no intermediates. The protein just almost immediately folds into its final stable shape, the native form. And then you can have proteins where maybe the native state has a very low energy state, but there could be an intermediate where there may be multiple intermediates that are significantly stable, which means that the protein can kind of fold into these intermediates and get sort of stuck, because maybe it takes some more energy to get them out of that intermediate state and sort of over this hump and then into the native form. Again, this is another way that proteins can be folded into or misfolded into these intermediate forms. So for those proteins, especially where they are commonly misfolded, then there are other proteins in most cells whose job it is to help those proteins fold into their native form. And collectively, these are called chaperone proteins. So a chaperone protein, just like a chaperone at a dance, just sort of makes sure the proteins are behaving. So again, folding intermediate hopefully ends up into its native protein, but sometimes that folding intermediate can misfold and either form an aggregate or one of these chaperone proteins. This is an example of a chaperone protein. It's actually a member of a bigger family of protein called heat shock proteins. That's why the prefix or that acronym HSP is used. So these are found in bacteria, and they are in fact found in cells, or they're expressed in cells when the protein or when the cell is subjected to heat. So heat again causes proteins to misfold, and so these proteins are turned on whenever the temperature of a cell gets above a certain level, and their job then is to find misfolded proteins and help them into their correct form. This particular one, the way it works is it finds so certain misfolded proteins or folding intermediates, and it doesn't fold them into their native form, but what it does is it prevents them from misfolding and sort of converts them back into the intermediate state. So you end up with, again, a misfolded protein getting converted back into its intermediate form using this chaperone protein, and then that just helps it drive toward its native state rather than being converted into an aggregate. And some of these chaperone proteins can be quite complex, so they can form these large complexes consisting of many different proteins. So again, this is a very important process in controlling protein function, making sure that the proteins in the cell are folding into the correct shape. And so this is just an example of one of those big giant complexes. So obviously protein shape is important for function, but protein folding or misfolding of proteins is also important in a lot of diseases. So one way that misfolded proteins can create problems is, again, one, the protein itself doesn't do its job, but some proteins, if they're misfolded, can self-associate. So that means that they can just end up with surfaces that allow multiple copies of the protein to sort of bind together, kind of in an almost crystallized fashion. And that leads to these structures called amyloids. So an amyloid basically is just a big tangle of proteins that are all of the same type that cluster together. Again, these amyloids can sometimes be cleared from the cell by proteasomes, but sometimes, especially with age, these amyloids can accumulate in certain tissues. And sometimes they can accumulate inside cells, sometimes they accumulate outside cells, but they usually eventually either kill the cell or cause some sort of functional deficit. So a good example of this is in Alzheimer's disease. So in Alzheimer's disease, these plaques form in the brain, and they eventually kill neurons, and that eventually leads to the symptoms of Alzheimer's. Another disease that's caused by protein misfolding is called prion disease. So prions are proteins that, again, they can misfold, but what they can actually do is cause other copies of that protein to also misfold. So they can not only, they themselves are not only malfunctioning, but they can cause other copies of themselves to also malfunction. And so in a way, they start to sort of spread, these prions. And eventually, again, they can do the same thing. They can cause brain damage. It depends on the tissue they're forming in. But prions actually are infectious. So if you eat meat, for example, that has a prion protein in it, and that protein is in your tissue, then you can then essentially catch that prion, and it can cause misfolding in your cells. This is commonly found, or the most well-known versions of this are a disease called mad cow disease. So the long name for mad cow is bovine spongiform encephalopathy. Basically, it means that you can see, like in this slice of brain tissue over here, little holes. That's why it's called spongiform, and it leads to eventually dementia and death. But there are human versions of this that again are caused by consuming meat or food that contains this protein in it. So we will stop there, and then we will next move on to the next chapter, which deals more with protein function.